Some of the more perplexing neurological diseases are being examined more closely with the advances being made in neuroscience. Among the most notorious of those diseases are Parkinson's, Alzheimer's, and more serious neuronal degenerative disorders. A novel correlation is being examined very closely, and that is the metabolic pathway involving iron in the brain compared to iron metabolism throughout the body. It has been suggested that the higher levels of iron in the brain are causally linked to these diseases. While this is a relatively new explanation for some of the root causes of these neurodegenerative diseases, it offers a new avenue for treatment, namely chelation. To be able to understand why iron is so important in neurological disorders, knowledge of the mechanism and purpose of ferritin is vital. The metabolism of iron begins with the complex ion formation of holotransferrin. This molecule is floating throughout the cytoplasm surrounding the cell, and once it has bonded with one or two iron atoms, the holotransferrin iron complex transports into a pocket of the cell membrane. Inside this pocket made of the cell membrane, a reduction protein reduces the iron to be used in other metabolic pathways. Once the iron has been reduced, divalent metal transporter protein, DMT, moves the iron inside the cell to be used by various proteins, such as the mitochondria. Iron concentrations in the brain increase later throughout life, and start increasing heavily in the 30s. As the levels of iron increase, ferritin store concentrations also rise. Ferritin is the protein that stores iron for use when there is a deficiency in the cytosol of the cell, and also serves to sequester it. Iron that is not immediately needed is stored as ferritin. Once inside the ferritin, the iron undergoes a conformational change so that it cannot escape. When a cell dies, the macrophages that are responsible for the cellular debris cleanup end up processing all of the released iron and themselves have high concentrations of iron. Parkinson's disease specifically affects iron-rich areas of the brain, such as the substantia nigra. Similarly, Huntington's disease is known to affect those iron-rich areas of the brain, and neuronal loss is observed. These observations, considered with the metabolism of iron in places such as the mitochondria, indicate that there could be a connection between the neuronal degeneration and the oxidative stress associated with having higher iron concentration in those neuronal areas. It is not clear whether or not the neuronal degeneration is a cause or an effect of the high ferritin concentration areas, but treatments based on those observations are being investigated. One such treatment is with chelation, which has been shown to work in studies investigating mice and has seen success in clinical trials, but further results will be needed to prove it beyond doubt. With these advances being made in the metabolism of iron, it seems likely that we are well on our way to understanding on a more fundamental and treatable level why neurodegeneration takes place. Through treatments such as chelation, many of the causes, such as in aceruloplasminemia, can be treated for at their source and the pathology can be improved. While the results do not indicate that there is a clear-cut solution, many of the suffering have a reason to be hopeful that they can once again live normal lives, and in the not-too-distant future may be able to receive a cure.